Welcome to Med in 5. The goal of our channel is to simplify medical concepts and help you understand them faster than a caffeine hit. This episode breaks down the Golgi apparatus, the cell's version of a post office. Give these questions a shot before we dive in. Which side of the Golgi apparatus buds off transport vesicles? What's a major role of this organelle in cartilage cells? What Golgi function involves the addition of sugars to proteins? Let's drop the brakes and roll. The Golgi apparatus modifies proteins made in the rough endoplasmic reticulum and lipids produced in the smooth endoplasmic reticulum. The modification process involves tagging the proteins and lipids with specific sugars or sugar chains through a process known as glycosylation. The sugar tags act like molecular labels that help the cell recognize where each molecule should go, whether toward the cell membrane, in the cytosol, or outside the cell, depending on the sugar type added. For example, mannose 6-phosphate tagging directs enzymes to lysosomes. For membrane renewal, the Golgi apparatus sends vesicles carrying lipids and proteins to the plasma membrane, where they fuse and replace worn components. The Golgi also acts like a recycling center for membranes within the cell, taking back membrane material in vesicles and sending it out again. The Golgi apparatus is made of stacked, flattened membranous sacs called Cisternae, organized into regions that process and ship molecules step by step. The cis face receives newly made proteins and lipids from the RER and SER through transport vesicles, which deliver these cargo molecules into the Golgi. Inside, the cis cisternae begin early modification and tagging. The medial cisternae carry out most of the chemical processing of proteins and lipids. The trans cisternae act as the final processing zone, where molecules are completed and sorted. At the trans face or trans Golgi network, the Golgi functions like a dispatch center, directing each product to its destination. Secretory vesicles are formed at the trans side to carry finished products to the cell membrane for release outside the cell or for use in other cellular locations. Different cells use the Golgi apparatus in special ways depending on what they need to produce and export. Plasma cells, which are antibody-secreting B cells, use it to package and secrete antibodies. Goblet cells rely on it for mucus production and release. Pancreatic acinar cells package digestive enzymes for secretion into the small intestine. Neurons use the Golgi apparatus to package neurotransmitters for signaling between nerve cells. Endocrine cells process and secrete hormones into the bloodstream. Hepatocytes or liver cells modify and export important plasma proteins. Osteoblasts or bone-forming cells use the Golgi to secrete collagen for bone formation. Chondrocytes or cartilage cells also release collagen to build the extracellular matrix of cartilage tissue. In spermatids, which are immature spermatozoa, the Golgi helps form the acrosome, a structure needed for fertilization. Melanocytes, the skin's pigment-producing cells, use the Golgi to package the pigment melanin into storage vesicles called melanosomes. Across all these cells, the Golgi acts as a customized packaging and shipping center, adjusting the products it releases, depending on the specific function of each cell. Let's go back to these questions. Which side of the Golgi apparatus buds off transport vesicles? Answer: trans face. What's a major role of this organelle in cartilage cells? Answer: extracellular matrix protein secretion. What Golgi function involves the addition of sugars to proteins? Answer: glycosylation. 0 slip-ups. You're ready for rounds. If this vid helped you out, support our channel by tapping like, share, and subscribe. Thanks for watching. Stay tuned to get the dose of Med in 5.